Hello everyone, welcome back to the Biotech Frontier. So in the last lecture, we have discussed about the PAGE, that is the Polyacrylamide Gel Electrophoresis, what these gel are made up of, and how the proteins are being separated on the basis of molecular size on these type of gels. We have also discussed about the basic differences between the SDS and the native PAGE. For today, we will focus upon the purpose behind performing the SDS as well as the native page. So let us start with the SDS page. SDS page stands for the sodium dodecyl sulfate. Sodium dodecyl sulfate, and page means polyacrylamide gel electrophoresis. This is the electrophoresis technique to separate the protein on the basis of its molecular weight. Let's suppose these are the proteins of different molecular weight, and we want to separate these proteins on the basis of what? On the basis of its molecular weight. So when we pass these proteins through the SDS page gel, that is these are the gels. We have already discussed about how these gels are made up of. So on the basis of the molecular weight, these proteins are being separated. So this one is the smallest one and this one here is the largest one. So the smallest protein separates faster because it moves faster through the gel because of its compatible pore size. All right. The next feature is that this type of SDS page, that is this type of electrophoresis, requires denaturing condition, means denaturing technique. It adapts the denaturing technique, means in this SDS page we are using such type of reagents, you can say, that those reagents can disrupt the non-covalent bonds. Non-covalent bonds means like the hydrogen bond or the ionic bond or the hydrophobic interacting bonds, not the disulfide bond. All right, not the disulfide bond, but it can disrupt or break the hydrogen bond, ionic bond, hydrophobic bond. All right, so these are the non-covalent bond. So here that particular reagent is the SDS. SDS is a reagent that disrupts the non-covalent bond. So this is the technique we are adapting in this SDS page. All right. And if we talk about that, why we are supposed to denature any protein, because we intend to study the protein, right? But for this special technique, we are supposed to denature the protein. Why we want to denature the protein? Because to get an accurate and consistent separation on the basis of its molecular weight. Because sometimes, let's suppose here, this is the tertiary structure of any protein. Tertiary structure of any protein. All right. So in our body, there are so many different forms of the proteins on that can like gives a protein to a different shapes, and to avoid the hindrance of the shape that may hinder during the separation, because here we are only supposed to separate the protein on the basis of the molecular weight, not on the basis of the shape. So because in our body the proteins are acquiring in a different shapes, they adapt different shapes in our body. Some are compact, some are loosely compact. So to avoid those things, we are supposed to linearize the protein. All right, we are supposed to linearize the protein so that there would be no discrimination. The second thing is that denaturation unfolds the proteins. Like suppose this is the folding state of the protein, but we want to unfold this protein to study each and every detail of this protein. If we want to study the inner part, intrinsic nature of the protein, so for that we are supposed to unfold these proteins into a linear polypeptide. So this is the linear polypeptide, and this one is the 3D shape of the protein, or you can say the tertiary or the quaternary structure of the protein. But if you want to study each and every detail of any protein, so for that it is necessary to convert that folded protein into unfolded state to get a linearized form. And this eliminates the effect of shape on migration. Suppose these are the two different proteins. If we linearize the protein, then there would be a no discrimination on the basis of the shape. That's why we are supposed to denature the protein in its linear form or in its primary form. There are so many other factors also, like to eliminate the structural variability. Means sometimes the proteins are having a multiple subunits. Let's suppose one subunit, second subunit, and third subunit. These three subunits whole together comprise a single protein. All right. So, and if we talk about any other protein, maybe the another protein is having only two subunits. So to avoid this type of structural variability, we are also intend to denature the protein. The second is the different conformation. Maybe these two are of having a different conformation. Denaturation ensure all the proteins are in uniform state, that is in a primary state, or you can say in a linear state or the linear polypeptide form. Another factor that are being considered here is to impart a uniform negative charge. Uniform negative charge is that the SDS itself is having a negative charge. So this SDS provides a coating throughout the protein. As you all know that the protein is made up of different amino acids. Some are positively charged, some are negative charged. All right. So if you want to migrate particular protein into any particular direction, all right, so there should be a uniform charge on a protein. Let's suppose this particular protein is having some positive charge, negative charge, positive, negative, positive, negative, because of having a variation in the amino acids. All right. So because of this, if I say to migrate this protein towards the positive cathode or anode, or the negative cathode or anode, all right, suppose you want to migrate this protein towards the positive side, but this positive amino acid will create a repulsion, and this negative charge will create a attraction. So this may denature the protein or disrupt the structure of the protein. So to avoid this, it is necessary that the whole protein must be having a single type of charge. So this SDS actually provide a coating on the protein. So if we convert this 3D form of the protein into a linear form, and then we treat it with the SDS, then this SDS page coats the protein throughout and gives it a uniform negative charge that is proportional to their length. Alright. And this masks the protein negative charge. So separation is based only on the size, not on the basis of the charge. So here again we eliminate the chances of charge variation. On the other side, if we talk about the native page, that is the native polyacrylamide gel electrophoresis. So this type of electrophoresis is basically used when we want to study the native nature of the protein. Native nature means the way by which or in which it is resides in our body. So the way it exists in our body, in the same form we want to study those proteins. So if we want to know how the protein behaves inside our body and what is the structure of the protein that when it was present in our body, so for that we need a native page. So the native page requires or it retains the protein structure in its native form. It also retains the protein activity as it was in its native state. It also retains or it also conserves the native charge as well as the conformation. So we do not need to hinder any of these. All right. So the separation depends on the size as well as the shape and as well as the chart. So these three factors on which the separation is based upon if we are using native page for the separation or for to study any protein structure or its activity. So this was all about the purpose of denaturing the protein in SDS page, and as well as why we are not denaturing the protein in case of the native polyacrylamide gel electrophoresis. So this was all about today. In the next video, we will discuss about the method of performing the SDS page.